Assalamualaikum warahmatullahi wabarakatuh. Good morning, good afternoon, and good evening for everyone. First of all, I would like to thank the committee for giving me the opportunity to present our research. My name is ADINDA NURAZIZAH from Universitas Pendidikan, Indonesia. I will present our study entitled Silico Evaluation of Curcumin, Quercetin, and Saponin as potential inhibitors of the adhesion of proteins EtMIC3 and EtSAG19 in Eimeria tenella. Before discussing the study, let me briefly introduce the research background. Coccidiosis is a parasitic disease caused by protozoa of the genus Eimeria. It is one of the most important diseases affecting the poultry industry because it causes severe intestinal damage, reduces absorption, decreases productivity, and ultimately leads to significant economic losses. Among all of the Eimeria species, Eimeria tenella is the most pathogenic one because it specifically infects the ceca and causes severe hemorrhagic lesions. As illustrated there, the proteins EtMIC3 and EtSAG19 play essential roles during the early stage of infection. This protein mediates the initial adhesion of the parasite to host cells, that allowing the infection process to begin. So far, the management of coccidia has frequently relied on synthetic anticoccidials, but we can see that prolonged of synthetic anticoccidial drugs has resulted in increasing drug resistance that's reducing their effectiveness over time. Therefore, alternative therapeutic strategies are urgently needed. Natural compounds have attracted considerable attention as potential alternatives. There are three components that we will discuss. First is curcumin, that we can derive from Curcuma longa. Quercetin that we can derive from many other plants, but one of them is Moringa oleifera, and saponin that we can derive from Sapindus harak. These three compounds have been reported to possess antiparasitic, antioxidant, anti-inflammatory, and immunomodulatory activities. However, as indicated in the research gap, there are still limited comparative studies evaluating these three compounds simultaneously against Eimeria tenella using an integrated in silico approach that includes a biological activity prediction, ADMET analysis, and molecular docking. The next one is the method. The study employed an integrated in silico research design. The ligand investigated were curcumin, quercetin, and saponin, while the target protein were EtMIC3 and EtSAG19. This study employed an integrated in silico workflow. We tried the ligands and the target proteins, predicted their biological activity and ADMET properties, prepared the molecular structure, identified the binding sites, and performed molecular docking, and finally analyze the molecular interaction. Now let us move to the first result. The slide presents the predicted biological activities generated by Pass Online. Table 1 summarizes the predicted biological activities of three compounds. Overall, all the three compounds demonstrated predicted antiprotozoal, anti-inflammatory, antioxidant, and lipid peroxidase inhibitory activities, which are beneficial properties for controlling coccidiosis. Among them, quercetin exhibited the most balanced, comprehensive biological activity profile. Curcumin showed superior potential for protecting intestinal mucosal tissue, which is particularly important because Eimeria tenella primarily damage the intestinal lining. Meanwhile, saponin displayed strong immunostimulatory activity, which is also known for its ability to disrupt parasite membrane integrity. These findings suggest that each compound contributes different biological advantage against coccidiosis. The next analysis focuses on ADMET profiles of the compounds. As presented in Table 2, quercetin exhibits the most balanced pharmacokinetic characteristics. It demonstrated good aqueous solubility, favorable intestinal absorption, appropriate tissue destruction, and the widest prediction safety margin. This characteristic make quercetin the most promising candidate from a pharmacokinetic perspective. In contrast, curcumin showed the highest intestinal absorption, that indicating good oral bioavailability. However, it was also predicted to interact with several CYP450 enzymes as both a substrate and inhibitor, suggesting possible drug-to-drug interaction. Finally, saponin showed the least favorable EtMIC profile. It exhibited very low intestinal absorption and was predicted to inhibit HERG2, indicating potential cardiotoxicity. Next, we evaluate molecular interaction through molecular docking. Table 3 summarizes the docking scores of each ligand against EtMIC3 and EtSAG19. Based on the binding affinity values, curcumin and quercetin showed their strongest interaction with EtMIC3 with the ID Q6R5NO, while saponin achieved its lowest binding energy with EtMIC3 with the ID D6VCP4. However, binding affinity alone is not sufficient to determine the best molecular interaction, as illustrated by the docking visualization on the right side. Saponin with B6PCP4 complex did not occupy the predicted binding pocket appropriately. The ligand adopted an unfavorable binding pose, suggesting that despite its low docking score, interaction may not represent a biologically relevant binding mode. In contrast, the saponin with Q6R5NO complex was positioned correctly within the predicted active site and exhibited a more appropriate binding orientation. Therefore, a Q6R5NO model was selected for subsequent interaction analysis of saponin and EtMIC3, even though its binding affinity was slightly less favorable than that one of B6VCP4. Overall, these findings emphasize that both binding affinity and binding pose should be considered when evaluating molecular docking results, as low docking score alone does not necessarily indicate the most reliable biologically meaningful interaction. To further validate the docking result, we analyze the hydrogen bond and hydrophobic interaction that form between the ligand and the target proteins. As shown in the left table 4 and 5, saponin formed the highest number of hydrogen bonds with EtMIC3. However, it did not form any hydrophobic interaction. In contrast, however, curcumin and quercetin form fewer hydrogen bonds, but also establish several hydrophobic interactions, resulting in a more balanced interaction profile. Similarity for EtSAG19 on the right tables, curcumin and quercetin exhibited both hydrogen bond and hydrophobic interaction, whereas saponin mainly relied on hydrogen bonding. Overall, this result indicate that a combination of hydrogen bond and hydrophobic interaction contributes to more stable ligand and protein complex. Therefore, together with the docking analysis, curcumin and particularly quercetin demonstrated more balanced molecular interaction with both target proteins. Finally, I would like to conclude our study. An integrated in silico evaluation demonstrated that curcumin, quercetin, and saponin all possess promising anticoccidial potential. Among the three compounds, quercetin showed the most balanced profile by combining favorable biology activity, pharmacokinetic properties, safety, and stable molecular interaction. Although saponin exhibited the strongest binding affinity, but its poor predicted bioavailability and potential cardiotoxicity limit its suitability as a drug candidate. Meanwhile, curcumin demonstrated favorable intestinal absorption and stable interaction with the target proteins, making it another promising candidate. Overall, this finding suggests that the tested compounds may inhibit the adhesion proteins EtMIC3 and EtSAG19, thereby interfering with the initial invasion process of Eimeria tenella. Future studies involving in vitro and in vivo experiments are necessary to validate this computational prediction. So I think that's all from me. Thank you very much for your attention, and I welcome any question or comments. Wassalamualaikum warahmatullahi wabarakatuh.